Hi, I'm Jonathan Beck with the Environmental and Food Safety Marketing Group for Thermo Scientific based in San Jose, California. In the experiment that I'm going to describe today, we took drinking water samples and spiked antibiotics into them and then analyzed them using the eQuant system and the Exactive Plus Orbitrap Mass Spectrometer. We chose about 20 different antibiotics that are commonly prescribed to humans and animals through veterinary or medical uses, and we spiked those into drinking water samples and analyzed them on the mass spectrometer. Traditionally, this analysis is done with triple quadrupole mass spectrometers, but in this case, we've decided to switch and use the Orbitrap for its full scan capabilities. Recently, antibiotics in water have become a concern in municipalities and drinking water supplies, as well as bottled water supplies, so we wanted to analyze these samples and see how much antibiotics were present in them. So we had a mixture of about 14 different antibiotics that we analyzed, and those are shown in this portion of the graph here in Table 2. And you can see the names of the common antibiotics that we analyzed here, ranging from some of the sulfa drugs to things such as carbamazepine, ketoprofen, and trimethoprim. What we did was spike our antibiotics at several different concentrations and quantitate the values that we would analyze using our online preconcentration scheme. Typically, these antibiotics are found at very low levels in the drinking water supply. And to analyze these on most mass spectrometers, we have to concentrate the levels up to a higher concentration so that we can see them on the mass spectrometer. Usually this is done using offline solid phase extraction. This process can be very time consuming and labor intensive. It's also prone to user error just from variances in operators or whoever is performing the analysis. What we do with our eQuant system is bring that online and automate it so that there is no manual intervention required. Everything's automated, it saves time, and there's less chance of having a mistake due to a human error. Our eQuant system uses two different columns as well as two different pumps coupled with the autosampler and the mass spectrometer, and it's shown in Figure 1 here. Basically what we do is we have a trapping column or loading column where we inject a large volume of sample, typically 1 milliliter, but we can go up to 20 milliliters. In this experiment, we injected just 1 milliliter. What we do is we inject that large sample of water directly onto the loading column. We then backflush that column. Our components that we are detecting are backflushed off of the column and onto the analytical column where we do a traditional HPLC or UHPLC analysis prior to introduction into the mass spectrometer. In this experiment, we spiked several different concentrations in the low parts per billion and low parts per trillion level for the antibiotics. You can see an example of one of the antibiotics, in this case sulfamirazine, at a concentration of 3 parts per trillion or 3 picograms per milliliter. The calibration curve is shown in the lower right-hand portion of the graph here, and here is the chromatogram of sulfamirazine at 3 parts per trillion. We get very good peak shape. So we're able to do very, very nice quantitation experiments using our full scan Exactive Plus instrument. Traditionally, this is the realm of the triple quadrupole instrument, but we've shown that the full scan capabilities of the Exactive Plus instrument coupled with the HCD collision cell that is present in the instrument can give you the same type of results that you get from a triple quadrupole experiment. Why would you want to use an Exactive or full scan Orbitrap instrument instead of a triple quadrupole instrument? Well, the nice thing about the full scan capability of the Exactive Plus is that you're collecting all the ions all of the time. What this allows you to do is go back at a future date if you need to re-interrogate the data and see if something was present that you weren't analyzing for at the time. You can go back, re-interrogate your data, look and see if a certain compound was present. It's in the data file. So you have that added flexibility of additional data mining techniques after the fact, which is something that's very powerful for users to have in their laboratory. Finally, on the far right-hand side of the poster, you'll see a screenshot of our software called ExactFinder. What we're able to do with the Exactive Plus is do full scan MS data at high resolution. We can then take that high resolution fragmentation spectrum and compare it to a library. And that's what's shown in Figure 4 of this diagram, of this poster. We're comparing a library reference spectrum to the spectrum that was collected on the Exactive Plus instrument. And the ExactFinder software will give us a rating score of how well those two spectra match up. That gives you the added confidence to say that you have really detected what you think you are looking for. It can also be used when you are doing unknown screening. If you're just running the sample, the software will suggest a possible match for that spectra and give you the results in the window there and say that it might be compound X, Y, or Z. Once again, we used online pre-concentration of a 1 milliliter sample of water spiked with antibiotics. We quantitated those levels to the low parts per trillion, and then we confirmed their MS full scan, high resolution accurate mass, Exactive Plus Mass Spectrum using the ExactFinder software. Thank you for watching this poster presentation today. For more information, please go to thermoscientific.com/ASMS, where you can download this poster or an application note. Thank you.